So what's the model for the movement of this S4 region during the voltage sensing of these voltage gate ion channels? So like we said before, if we think S4 region being alpha helical domain of these ion channels with this positively charged residues, either arginines or lysines, okay, what you can think is that this whole region, if it's alpha helical, it could move in response to the polarization towards the extracellular side of the plasma membrane. And again, when the cells then hyperpolarize, then this uh, alpha helical domain could uh, go back to their resting state. These movements of this S4 domain or S4 region of these ion channels somehow are able to couple with of this gating mechanism to open the permeation pathway or the pore of these ion channels and then allow the flux of ions. So there are a lot of experimental evidence to support that kind of model of alpha helical S4 domain generating the gating currents and the activation process of these channels. Uh, Somewhat unexpectedly, people start to develop another model based on mostly by X-ray crystallography or X-ray crystallography when it was performed in voltage-gated potassium channels. Uh, so it's very difficult to crystallize potassium channels or any kind of channel because this type of proteins have these hydrophobic um, regions which makes very difficult to extract these proteins from this hydrophobic environment. So you have to use strong detergents. In that case, what happens is that you don't get good crystals of these proteins. It's much easier to get good crystals of proteins that are very soluble so crystals that are very globular or intracellular localized proteins are much easier to get good crystals. But for ion channel has been very difficult to get good crystals. Uh, so what people start to do is to clone and to express large amounts of potassium channels or other types of channels from bacteria. So for example, here is a bacteria potassium channel called KVAP potassium channel, and we know this is a potassium channel because if you clone this channel from bacteria and then you express in a mammalian cell or you put in a lipid bilayer, it can generate potassium currents. And furthermore, if you align the sequence of the KVAP channels with, for example, a shaker potassium channel, as shown in this figure, you can see there are a lot of sequence similarity. So for example, in the S1 domain, you see a lot of amino acids are very similar. Same for S2. And S4, you can see this voltage sensing region here have these positively charged amino acids. And finally, you see the other domains S5 and S6. So this KVAP potassium channel is a bona fide potassium channel. And the advantage of working with these channels was that it was easier to get good crystals of these channels, and therefore with crystals, people are able to then obtain high resolution structure of these potassium channels. And what people saw, and if you get crystals of these channels either in the closed or in the open state, is very many changes in terms of structure. And in particular, they are very interested to see what kind of structure changes happens during when you go from the closed to the open state. So what they saw was actually a model that's not entirely consistent with what we said before of alpha helical domain moving up and down across the plasma membrane. Instead, the S4 domain work together with a part of S3 domain, okay, called S3b domain, and this whole region between S4 and S3 would form this macromolecular structure that we call a petal. This petal would contain this positively charged amino acids that are able to sense changes in transmembrane voltage. And this structure now doesn't move across up and down across the plasma membrane, but instead it would move more like a petal, moving from the intracellular domain to a more oblique kind of configuration, and this kind of configuration would then be responsible to to, uh, to transition these channels from the closed to the open state. So there's still a lot of debate to see which model accounts better for some of the experimental evidence obtained using physiology experiments and so on. And so therefore there's still somewhat debate about which model is correct.